Population scale datasets are critical for identifying pathogenic and somatic variants because with the reference alone, while we can identify most of the variants in a sample, we can't determine which of those is driving disease. And even with effect prediction algorithms and databases like OMIM and ClinVar, we still don't identify the deleterious variant in most patients. While we can't find it, we do know something about this variant. In particular, it's under purifying selection and should be rare. Therefore, if we can assign an allele frequency to every variant in a patient, then we can ignore all of those variants that are more common than their disease. With a database as large as gnomAD, that means we can reduce the number of variants that we must consider by 2 orders of magnitude. Unfortunately, this really only works with single nucleotide variants, where for every sample and every aligned read, we look at every position in the genome. The completeness of this analysis and the homogeneity of the data give impressive precision and recall results. If you're not impressed by this, check out the scale of the y-axis. As a result, we get nearly complete and error-free catalogs of small variants in these populations. SVs, things like duplications, inversions, and deletions, are a different story. First, we have multiple signals we must integrate across, and some of those, like read depth and discordant reads, don't provide direct evidence of the variant. Split reads do provide sequence-level evidence of the variant, but they are often confused by local homology. The result is that we must make difficult trade-offs about the true positives and false positives in our SV catalogs. And given our general distaste for false positives, we often use aggressive filters. The insidious result of those filters are many false negatives. And a false negative is a killer for an allele frequency reference. Just consider the case where you have a patient who has a variant that is not in your database. For a database like gnomAD, you can assume that given the completeness, that variant is ultra rare. For an SV catalog, you don't know if that variant is rare or if it has been filtered. Long reads do improve performance, but they are still far from the performance of short read detection. So, what do? With our structural variant index, STIX, we avoid the detection problem altogether and turn this into a search problem, where we take a population of aligned reads, we remove all of the normally aligned reads, and index just what remains. For a given variant, this becomes a query where we find all of the alignments that agree on the existence of that variant in that sample. For this query, for example, we see that there is evidence in multiple samples and interpret that the variant exists in the population, whereas another query finds no evidence supporting the existence of that variant in this population. While we don't get rid of the false negative issue altogether, I argue that we drive it to the sensitivity of the data. Had we sequenced more, we might have picked up evidence of that variant. How do? Before we get to it, we're going to start talking about somatic variants, and we're going to assume that they are rare. While this might not always be true, it is generally true, and it's especially true for those variants that are driving tumor formation and cancer evolution. Our experiment looked at the 181 prostate cancer samples from PCAWG, and we looked at how population-level filters compete with the gold standard of calling somatic variants based on tumor normal pairs, and you return just those variants with evidence in the tumor. We looked at 3 population filters where we first called variants just in the tumor sample, then removed any variant found in the population. The 3 methods were a STIX index of the 1000 genomes cohort, the 1000 genomes cohort SV catalog, which is based on the exact same data that STIX looked at, and then the gnomAD SV catalog. And just as a reminder, 1000 genomes had 2,504 individuals, and gnomAD had about 6 times as many, about 15,000. Here are the results. So when we call variants on tumors alone, we find on average 3,892 deletions. We have done this also with duplications and inversions, and the results are very similar. We then look at filtering. The first filter being a tumor normal paired calling, in which case we find 51 false positives. And I should note here that the truth set is the published PCAWG calls. STIX, impressively, has fewer false positives, about 30% fewer. For the other population filters that are based on SV catalogs, you see the impact of these false negatives. 1,000 genomes has 2 orders of magnitude more false positives because there are so many false negatives. Nomad, which has 6 times the number of samples, doesn't even cut this number in half. So while adding samples can help the false negative issue, I argue that we can't outgrow our databases to account for the false negatives. Interestingly, the true positives are fairly consistent for all methods, but you must consider that these true positives are mixed in with the false positives, making them hard to identify in the population level filters. Now there's something interesting going on with false negatives. All 3 population filters have, on average, 7 more false negatives. That is, there are 7 variants that are in the population and in the PCAWG call set. Just a quick recap, STIX is simple. I wasn't explicit about this before, but there is no SV calling, there is no SV merging. If you have ever done this for a sufficiently large cohort, you know how difficult, complex, and fraught this process is. You simply index the cohort, and then everything is a search. It's also quite powerful. It's as good and possibly better than the gold standard tumor normal calling for somatic variant identification. It's also very fast. Once you've indexed, which the indexing time is proportional to the size of your database, once you have that index, queries take about a second, and this is an embarrassingly parallel problem that can scale to very large call sets. Since this is search, it's flexible. We can have indexes for multiple populations. We had been looking just at germline indices, but we're going to start exploring tumor-specific indices and the integration of those different populations. And we can adjust how we query so that it matches the question. We'll explore these last 2 points in the following slides. The ERG Tempest 2 fusion is one of the most common variants we observe in prostate cancer samples. So given the standard query for STIX, if we observe a patient with this variant and we run the query, we convert that to query regions and find that 6 samples have a matching query, which is a little bit surprising given how prevalent this fusion is. But we aren't necessarily bound to these query regions. We're currently exploring the idea of a functionally equivalent query where, in the case of fusions, that query spans the entire gene body and will identify any variants that connect these 2 genes. In this case, we find 130 samples, which is actually a bit more than PCAWG found. Following the functional equivalence, more broadly, we wanted to look at fusions across the entire cohort. So we first called variants using our tumor normal approach, and we found 1,335 unique fusions. To figure out which ones might be driving disease, we first looked to see which are most abundant in the prostate cancer samples using our new STIX prostate cancer sample index. And here you find a bunch of duds. You find nested genes, seg dupes, read-through transcripts, and duplicated genes. And this result is actually backed up by looking at their frequency in the 1,000 genomes. So now we're integrating 2 different indices, a disease one and a germline healthy one. What makes more sense here is to look for things that are rare in 1,000 genomes and frequent in the prostate cancer samples. And the gene list contains all the heavy hitters for prostate cancer. There are a few surprises in here, genes that we had not associated with prostate cancer, which we're following up with some collaborators now. While STIX can really improve your call set, the recent history of structural variants have led to some trust issues with these call sets, and it's hard to avoid manually inspecting our variants. And for most of us, myself included, that means IGV, a super powerful and super general tool that can require some local customization, which becomes a bit tedious over time. To help with SV manual curation, we created Samplot, which is SV specific and is intended to be looked at for about 1 second. We accomplish this by highlighting all of the signals that are critical for curating structural variants, like coverage and the alignments and the size of those alignments as they correspond to the size of the variant. Samplot can look at just about every sequencing technology, short reads, long reads, phased reads, and multiple samples at once. But even looking at multiple samples, you have a narrow view of this variant, which can lead to false negatives and false positives. So our idea was, why don't we bring Samplot and STIX together for a population scale visualization, which would allow us to go from a case where we thought it was a slam dunk somatic variant, clear signal in the tumor, no signal in normal, yet looking at this variant in the context of 1,000 genomes, we find this is a false negative in the normal, which could have led to a false positive somatic call, if not for the population evidence telling us this variant is common in germline healthy samples. That's it. All of our software is freely available on GitHub. This work is led by a staff scientist in my lab, Murad,